Hello, chemist and biochemist. In this video, what I would like to talk to you about is the polymerase chain reaction, also known as PCR. I want to talk to you about all the different components that are involved, as well as some of the tools that you have to have in order to do the polymerase chain reaction. Now, the first thing that I'd like to share with you is I'd like to identify the person who is largely credited with the discovery of PCR, and that's a fellow by the name of Cary Mullis, K-A-R-Y Mullis. Cary Mullis was awarded the Nobel Prize. I believe that it was in chemistry in 1993 for his work on PCR. He died just a few years ago. I believe in 2019 was when he passed away. His background and training was as a chemist, and he happened to be working at a corporation known as CETUS, CETUS, in the early 1980s and did lots of pioneering work on um, amplifying DNA. Now, PCR arguably is maybe one of the most significant life science discoveries of the past 50 years or so. And basically, it takes one piece of DNA, identified here by the single line, one piece. And from that, you can make basically Xerox copies or perfect copies. And you could go ahead and say 1 million copies, 1 million error-free copies of that same piece of DNA. Now, what the polymerase chain reaction consists of is, first and foremost, you have to have a piece of DNA that you want to amplify. Now, that double-stranded piece of DNA, dsDNA, is held together by hydrogen bonds. And so the very first step in PCR is to take those double strands, those two strands, and separate them from one another. Now, when you separate them from one another, you end up with two single-stranded pieces of DNA. In order to get there, in order to get that separation, to break those hydrogen bonds, you have to put a lot of energy into it. You have to heat it at about 98 degrees Celsius. When you heat that DNA at 98 degrees Celsius, that dsDNA separates, as we demonstrate here. The next thing that you're going to do is you're going to cool this DNA to about, let's say, 55 degrees Celsius. And at that temperature, your two pieces of DNA, your two single-stranded pieces of DNA, will have a complementary region, a synthetic complement that will bind to each one of them. So this step is where your primers bind. Now, where those primers bind is, well, you're going to have basically a piece of your single-stranded DNA is going to have a region that is double-stranded. Where those primers are going to bind together or bind with your single-stranded piece of DNA, this is also known as hybridization. Now, once you have these pieces of DNA that a portion is double-stranded and a considerable portion of it is single-stranded, what you're then going to do is you're going to heat your sample up to 72 degrees Celsius, roughly. Now, these are plus or minus a couple of degrees. All of these temperatures, 98, 55, 72. But they're all going to be within that range. And at that point in time, when you've heated your sample up to 72 degrees Celsius, you have these pieces of DNA that one short region is double-stranded, and then one portion of it is single-stranded. That is where you're going to have, and I'm going to have this lima bean shaped structure. This is going to be your enzyme. The enzyme, the TAQ polymerase, is going to bind and begin synthesizing a completed double-stranded piece of DNA. So it's going to see that single strand and it's going to say, aha, I see an A nitrogenous base. So I need to add a T. I see a C. I need to add a G. and basically fill in those gaps. So this step with the Taq polymerase is also known as your polymerization step. So we basically have three very simple steps. 1, 2, and 3. First is our, you'll sometimes see this called denaturation. You might also see it referred to as melting. Your second step is your hybridization or your annealing. And then your third step is your polymerization. Oopsies. Polymerization. Or you might also see it referred to as your synthesis. So denaturation, double-stranded pieces of DNA separate. Annealing, single-stranded pieces have template, or you have these primers come in and bind. And then your polymerization, where you're synthesizing and completing this complementary piece of DNA. So these three steps are going to be repeated over and over and over again. You're going to take one piece of DNA, and at the end of the first round, you're going to have two pieces of DNA. At the start of your second round, you're going to have two pieces of DNA that you're going to go on to be 4 from that. So you're just going to you're going to continue to repeat this over and over and over again. Now, some of the components that you need to have are, you need to have this Taq polymerase. This is a DNA polymerase that is a thermostable polymerase. So this is an enzyme that can endure heating to 98 degrees Celsius, 55 degrees Celsius, and 72 degrees Celsius. You then also need to have these synthetic oligonucleotide primers. Now, these are pieces of DNA that have been generated that will complement a short portion of your template DNA. They're not going to be exact duplicates, because if they were, that's kind of the point of using PCR to amplify a piece of DNA. These are short. These are, anywhere from 16 base pairs to maybe 30 base pairs. And these are used when you're trying to amplify a piece of DNA that's, say, 600 base pairs long. So you use these small pieces of DNA that anneal to or hybridize to another part of of your template. Now, one thing that's not on here but worth noting is, in order for your enzyme to work properly, you have to have magnesium 2+, the divalent cation magnesium, and kind of the most important part is, well, the funny thing is these are all very important parts, but one of the most important parts is you have to have DNTPs. You have to have your deoxynucleotide triphosphates. What that means is DATPs, DCTPs, DGTP, and DTTP. Because here, if we go back to our annealing step, when we go from our annealing to our polymerization, when we're adding in a new base, what we need to be pulling from is a pool of DATP, DCTP, or DGTP, or TTTP. If you see an A, if the enzyme sees an A on the template, it needs to pull a DTTP. What it will do is it's going to take DTTP, and it's going to make DTMP, monophosphate, and PPI, or inorganic pyrophosphate. And this DTMP is what's going to be added to our piece of DNA, our newly synthesized piece of DNA. So these are basically the steps and the components: denaturation, annealing, polymerization. At that polymerization step, you're actually synthesizing that new piece of DNA. Nowhere before then are you actually doing that. So what temperature is the synthesis taking place at? About 72 degrees Celsius. You have to have your template or your primers bind to your template. That's going to happen at about 55 degrees Celsius. How are you going to be adding that? Well, DNA polymerase from thermophilic bacteria like Thermus aquaticus, that Taq polymerase, is going to use magnesium in these primers and all of your deoxynucleotide triphosphates. Well, I hope that was a helpful way of thinking about PCR and the polymerase chain reaction. I hope that if you can kind of hone in on one thing, you can understand this process right here, where we're going from one piece to two complete, or one double-stranded piece of DNA to two completely double-stranded pieces of DNA. All right, well, have a good one.